Hello, everyone. Welcome to our final session of the day. I hope everyone has enjoyed the earlier panels. If this is your only session today, welcome to the International Research Mobility Symposium. My name is Sylvie Jasen, and I'm the International Projects Coordinator at Carleton International. I have had the pleasure of assisting two of the speakers in this panel in their research visits under mobility programs just this past year. We have three speakers in this final panel. Each presentation will be 10 minutes, and we will have time for question and answers afterwards. You are welcome to pose questions and comments throughout the presentations in the chat. These will be monitored and collected and then addressed once all the speakers have finished. During the Q&A, you will also be able to raise your hand. That means your virtual hand in the Zoom, and our tech support will unmute you to ask questions if you would prefer to do it that way. Now for the presentations. So we have a diverse group here today. All three are PhDs in very different fields. They will each outline the research they conducted during their research visits, either at Carleton or abroad, and then speak to the intercultural experience that informed their work. So our first speaker is Alexandra Lamb. Alexandra is a PhD student in public policy at Carleton University. As well as a student, she is a junior policy analyst working for the federal government in the Indigenous Affairs Directorate within ESDC. She completed her master's degree at Carleton University in the Masters of Philanthropy and Nonprofit Leadership program within the School of Public Policy and Administration. And the research she is presenting here was her capstone project. She was fortunate enough to be connected with Dr. Sheila Cannon from Trinity College in Dublin, Ireland, where they worked together on exploring social movements, namely the abortion referendum. Alexandra, you have the floor.
Great, thank you so much for that introduction. So today I'm actually going to do it opposite. So I'm going to start with my experiences in some of my time in Ireland, and then I'm going to jump into the research that we were able to do. And so the team that I worked with was Dr. Sheila Cannon from Trinity College. She was my visiting supervisor, and then Paloma Rago, she's my supervisor now in my PhD as well at Carleton University, and I couldn't have done it without either of them. They were both extremely supportive throughout the whole thing. For anyone who hasn't had the opportunity to travel or study abroad, it's an incredibly enriching and invaluable experience. You learn a lot about yourself as well as the world. Specifically in Ireland, I had the opportunity to work with such diverse scholars, academics, people from the community, activists. I got to see, well, it's an island, so there's lots of beautiful scenic areas that were actually really crucial to the whole process because it gave me a lot of time to do a lot of self-reflection, which I think is very important for people in general, but specifically for academics and researchers to really understand what we're learning, what we're experiencing, and how we're processing that internally. And that helped me a lot with my research. So I became much more open to differing perspectives, especially because our topic was very highly sensitive and polarized. So this was really important for me, and that is what I learned abroad, to be open, to accept new perspectives, and to just be willing to learn what other people have gone through. So we'll go right into the research. And so our study focused on civil society organizing leading up to the referendum and how activists managed to move away from the debate, highly polarized debate. And essentially it was emotive deadlock. And it moved people to vote to remove the abortion ban. So the main literature we looked at was organizing and organizational processes, issue framing, and valence. And as you can see, our main research question was, how do activists organize and mobilize public support around highly polarizing issues? Now, for people who don't know, the Eighth Amendment, so from 1984 to 2018, the Eighth Amendment of the Irish Constitution was essentially a ban on abortion in Ireland. It was providing equal right to life for the fetus as well as for the pregnant woman, with a 14-year prison sentence for breaches of this law. So it was essentially a 35-year ban on abortion. and then in 2018, it was finally repealed. So as you can see from the timeline that we created, just how long this movement really is and how complex it became. And this is really why we were interested in this. And we wanted to know why it was so successful this time around, with a 66.4% of people voting for yes to repeal the 8th Amendment. So we'll go to the next slide. So here I will talk a little bit about the story. And this story sort of carries a legacy of shame and silence about unwanted pregnancies, and sort of had this stereotype of who was really getting these abortions. And typically it was this very stereotypical image of a young girl getting pregnant out of wedlock. And so there was a lot of shame and guilt. It was highly emotive and very, very polarized. You can, if you know much about Ireland, there was a lot of religion that was playing into this. So it was very almost taboo to talk about in the public. This amendment did have a lot of impacts for many women, some women seeking the service elsewhere, such as England, for example. And there was many cases, Miss C, Miss X, Miss Y, that's how they were labeled, which brought a lot of awareness to the severity of this issue. For example, in 1992, the X case was a 14-year-old girl who was banned from traveling to end a pregnancy, to get an abortion, that was a result of rape. Now, she became suicidal, and she appealed to the Supreme Court. And this was actually really fundamental because it resulted in a referendum allowing for access to information for people regarding abortion, access to travel to get an abortion, and also suicide as a grounds for abortion. However, the most influential case that we looked at was Dr. Savita Halappanavar, and you can see her in the photo there. So in 2012, she did pass away at age 31 from septicemia, which is an infection that she contracted after she was denied an abortion during a miscarriage that she was having. And this set off outrage across the country, which really gave momentum to a growing call for change. The death of Savita Halappanavar was mentioned by every single person that we had interviewed as an event that really changed the debate on abortion in Ireland. Now, while stories of women's experiences of abortion had emerged before, following the death of Savita Halappanavar, there was this outpouring of stories on multiple outlets. So news, debate programs, radio, television, newspapers, social media platforms. It sort of blew up after that. But the two specific initiatives that we looked at the time were Together for Yes, which is a campaign that had over 100 organizations, and it was very strategic and experienced. And they conducted focus groups and tested out language to really understand what would reach the public. It became much more about this emotional framework opposed to this rights-based framework, which had typically been in Ireland historically. So, and then the second one that we looked at was In Her Shoes, which is a Facebook group, and it was very spontaneous. It was sort of organic. It was a safe way for women to tell their stories. It was created with the sole purpose of providing women with an anonymous platform to share their lived experiences without fear of retribution. This page was founded and maintained on the Pillar of Compassion. So this is much different than previous attempts to get stories out there. And it really showcased this new narrative of openness and acceptance. So I'm just going to quickly go over the methodology. It's pretty clearly outlined, but our method was process tracing, and we looked at the time period between October 2012 and May 2018, when the referendum was appealed. And our data collection strategies were mostly qualitative in nature. We did go through some archives. We went to rallies, book premieres, events. We looked at social media platforms, web pages. It was very exciting to do that sort of research. And then we used NVivo for coding. So we looked at organizations, key individuals, critical juncture periods, specific emotions, types of storytelling, that sort of thing. And we can go to the next slide. Okay, so here I'm really going to talk about what our key findings were. And I think I have about 2 minutes left, so I'll try to make it brief here. But what we really wanted to showcase here, and our central argument essentially, is that we show how harnessing emergent frames in narratives in organizing processes can create public mobilization, organizational and social change, despite these really highly polarizing issues that are at hand. And as you can see here in the diagram, we have the two initiatives that I was talking about before. So we have our Together for Yes, which we put at the top in this strategic framing bubble. It's very informative, they're mobilizing, it's very thought out. And then at the bottom, we have In Her Shoes, which we put in the emergent messaging and emotive storytelling bubble. And this was very organic. It's all about lived experience. It was created essentially just for a few people, and it ended up being countrywide, highly adopted way to tell stories. And so what we talk about here, because primarily in the literature of issue framing, you typically hear about the strategic, very experienced framing. And that's why we have the dotted line going to social change. So because it can happen, social change can come from just being strategic. However, in this specific context and in instances where it's very complex social issues, we see this middle frame here that we called responsive framing. And this is where the strategic and the emotive, the emergent messaging come together in the middle, and they create this sort of responsive framing, this sort of iterative, bottom-up, actor-centered approach to social change. And that's where we saw the real change within the movement. So this is sort of our diagram of process of mobilization and social change. It is a case study, so it's very specific to Ireland and the abortion referendum, but this is responsive framing is our theoretical contribution. So that is all I have for you guys today.
Great, Alexandra. Thank you so much. That was very interesting. Some questions came in, but we'll save those, like I said, until everyone is done speaking. So our second speaker is Gayathri Subramanian. Gayathri is a first-year PhD student at Northwestern University, Evanston, Chicago. She is working in the brain, behavior, and cognition area of psychology. Her work revolves around memory consolidation, psychophysiological measures, and concealed information. Prior to her PhD, she graduated from SRM Institute of Science and Technology, Chennai, India, completing her undergrad in biotechnology engineering. Her research experience involved studying the interaction of HDAC and tau protein, leading to neurodegeneration in the Drosophila melanogaster. And for her undergraduate thesis, she worked in Carleton University in the Abizade lab studying Ghrelin and female metabolism. Gayathri, you have the floor.
Thank you, Sylvie, for the introduction. So I am Gayathri, and I'm a PhD student in the brain behavior and cognition area at Northwestern University. I did my undergrad in SRM, as Sylvie had mentioned, and I came to Carleton for my undergrad thesis, and I worked with Dr. Alfonso Abizade, and we studied the effect of chronic Ghrelin administration in female mice. So the problem that we wanted to tackle with our study was obesity, which is a major health epidemic. And one of the reasons our focus was on women's metabolism is because of the fact that 40% of the women in India and over 50% of the women in African American countries, as well as the Caribbean countries, are affected by obesity. And it's becoming a major health epidemic that's sort of taking over even more so than people, malnutrition and underweight people. So we wanted to tackle this problem. And one of the ways we thought we could do that was by using Ghrelin, which is a peptide hormone that's secreted in the enteroendocrine cells of the stomach lining. So it is a hunger hormone, and it is responsible for regulating feeding behavior, appetite, fat accumulation, as well as the general homeostasis of the body. So how we wanted to do the intervention, how we wanted to study if there can be any possible interventions using Ghrelin was using female mice. So we had about 39 female mice that we observed over a period of 3 weeks, we had observations before treating them with ghrelin, and we had observations after treating them with ghrelin, to see how ghrelin affects the metabolism. So there have been a lot of studies that involve ghrelin administration in males, and also having more acute ghrelin administration where injections are given and immediately after 2 hours the effects are studied. But I believe that this is one of the few studies that involved chronic delivery of ghrelin, that is continuously over a period of 3 weeks. And we used a control group where we treated the mice with saline, and we had a treatment group where we administered ghrelin through 2 ways. We used a peripheral administration and we used a central administration. So peripheral, we ensure we delivered the ghrelin at a more subcutaneous layer near the stomach. And for central administration, we delivered the ghrelin through the brain. So we calculated the amount of exogenous ghrelin to give in such a way that it produces an effect, at the same time does not have 2 adverse effects in the mice. So we had 2 doses for peripheral. We had 10 and 40 micrograms. And for central, we had 4 micrograms. Next slide, please. Yeah, so the experimental procedure that we followed was initially we took baseline measurements, body weight, we took how much they're eating, the food intake, as well as vaginal lavages. Since we worked with female mice, it was important for us to see how the hormonal effects are present in the form of their estrous cycle. So mice go through a 4 to 5 day estrous cycle. So we wanted to see if ghrelin was having an effect on that. And so vaginal lavages ideally mean that we're going to be taking the cells, the vaginal cells, and we're going to be analyzing them to sort of decide what day of the estrous cycle that they are in. So this baseline measurements went on for about 20 days, because as soon as the mice came to the lab, we have to let them adapt to the circumstances and the environment, and then we go on to taking the measurements. After 20 days, we did the surgery, and we inserted a cannula in the lateral ventricle of the brain to provide the central administration. And then we inserted a mini osmotic pump in the subcutaneous layer near the stomach to provide the peripheral administration. And for a period of 14 days, we had the post-op care for the mice, as well as take post-treatment measures. So we again took their body weight, food intake, and vaginal lavages, but now they're obviously been treated with either saline or ghrelin. So we hope to study if they have any effect on the mice. And once we did that, we also wanted to test how ghrelin is affecting glucose clearance. So obviously, if metabolic disorders like PCOD and diabetes and gestational diabetes, one of the main causes or involvement is glucose. So we wanted to see if ghrelin has an effect on glucose and insulin as well. So we basically ensured overnight the food was taken away from them overnight, and the next day they were measured for the baseline glucose clearance, how much glucose they have in their bloodstream having not eaten food. And then we gave them a 20% glucose injection, and then we took the glucose levels over a period of 15, 20, 30, and 120 minutes. Then we proceeded to place them in a metabolic chamber, which is basically a phenomaster chamber in which the rate of change of the weight, as well as how much they're moving based on infrared technology, was observed for over 48 hours. So both the glucose clearance test as well as the metabolic chambers took place over a period of 7 days. Once we were done with that, we euthanized the mice through live decapitation. We proceeded to collect their brain tissues, ovaries, fat tissues, etc., to see how, apart from these behavior measures, how is ghrelin affecting them at a more cellular and molecular level. For those kind of studies, we had to euthanize the mice. Lastly, we did data analysis, wherein we basically plotted graphs to see how much weight change was experienced or how much they were eating, and also the percentage of estrus days. So the way we analyzed the vaginal lavages data was count the number of estrus days. So estrus days are ideally the days in which the estrogen level in the mice is the highest. So we wanted to see if there was a direct interaction between ghrelin and estrogen. So we took the percentage of estrus days as in the data analysis, and obviously we analyzed the glucose clearance data as well as the locomotor activity to see how much how do they increase their movement in response to ghrelin, so that if ghrelin has an effect on calorie burning and all of that stuff. Next slide, please. So these were the results. I have included some of the significant results that was there in our study. So the weekly weight change was trending towards interaction, especially in the first 2 weeks in the peripheral group. And as you can see, the trending towards interaction was between the 2 doses of Ghrelin in the peripheral group. But towards the 3rd week, you can see that both the control as well as the treatment groups all have pretty much similar weight change. So in the peripheral group, it was kind of observed that Ghrelin's effects were fading over the 3 weeks. And the average locomotor activity also seems to have not that much difference between the treatment and control group, both in the light cycle as well as the dark cycle. And then in the cumulative food intake, or as well as the average food intake, it was seen that there was no significant results in the peripheral group, but in the central group, there was trending towards interaction, as well as significance in the 3rd week of how much they were eating. So the mice that were treated with Ghrelin centrally seem to have had a significant increase of the food intake. And they also, the present in the central group did not really experience a fading of the Ghrelin's effects. And the analysis of the glucose clearance test, we basically plotted the area under the curve. And so ideally, the relationship is that the more the area under the curve, the lesser clearance of glucose. And clearly for both peripheral as well as the central group, the control the most area under the curve, meaning that the glucose in their bloodstream was the highest. Whereas the group that was treated with Ghrelin seems to have cleared the glucose better because Ghrelin has an effect on insulin. So this led to clearing of the glucose in the bloodstream. Lastly, the analysis of the percentage of estrus. So in all of the literatures that have been done so far, Ghrelin and estrogen have sort of like a counter-interactive ability. So the more the estrogen, the lesser the Ghrelin, and vice versa. But in our study, it was kind of counterintuitive because both the peripheral as well as the central group, the Ghrelin-treated mice seems to have more estrus days than the control group, which is, as I said, counterintuitive to a lot of different literatures, meaning that there seems to be a sort of direct interaction and proportional interaction between estrogen and Ghrelin. So overall in this study, it was kind of observed that the food intake of the mice did increase because of Ghrelin, but their calorie burning, that is seen through the average locomotor activity and how much amount they're increasing their activity, did not seem to increase. And the weight change was also not significantly different from the start of the experiment till the end of the experiment. All of this kind of goes to say that there is some kind of metabolic storage happening. And since we know that Ghrelin has an effect on fat accumulation, we hope to sort of analyze their fat tissues to see if the exogenous Ghrelin that we're supplying is leading them to accumulate the calories that they're eating instead of burning them off. But at the same time, they don't seem to have any significant weight change. So we want to proceed to see how the balance between the endogenous and exogenous Ghrelin is by analyzing the bloodstream Ghrelin as well as the gene expression changes in the brain. So I was there in Carlton for the behavioral part of the experiment, which was till this point, post which my colleagues in the Abizaid lab took over to see the more cellular and molecular aspects of the study. Next slide, please. So I just wanted to emphasize the various advantages that I found that the research mobility that I experienced had. So these are some of the categories that I thought of, which was essentially the research practice. Coming from India, I had a very different educational system, and I got to experience how the educational system and the curriculum and the way research was being done at Carleton. And this led me to understand the different kinds of ways of research and helped me adopt new techniques. And at the same time, I was able to inculcate certain techniques in the lab as well. And I believe that the replicability crisis, which is going on in the field of science, even though not prevalent in neuroscience, can definitely be decreased through this method, because uniformity and exchange of research practices can definitely help decrease the replicability crisis. Obviously, a good intercultural experience. I got to meet so many new people, and I was able to expand my cultural knowledge. I got to experience the Canadian lifestyle. I got to celebrate Easter and Groundhog Day and all of those wonderful Canadian holidays. And at the same time, I remember taking a lot of my lab friends to eat Indian food. So I was able to share a bit of my culture as well. And obviously the new experiences that one would gain out of such an intercultural experience is coming from a tropical country. I got to experience snow and the winter wonderland of Canada, and doing ice skating, snowshoeing, etc. So basically gave me a holistic experience, which not only was about academics but also learning new culture, and it helped me meet new people, make new colleagues, etc. And obviously I was there during the second part of my stay was when the COVID outbreak happened, so it became quite an adventure for me traveling back. And the interdisciplinary research, obviously because I came from an engineering background and the people in the lab were from a neuroscience background, there was a lot of exchange of knowledge and differences in specializations. This led to collaborations and collaboration of distinct areas of science, and this, leading to my next point, led to new ideas and novel research ideas and experimental designs, because people from different educational backgrounds bring different perspectives to the table. This led to more brainstorming and coming up with new ideas. And finally, my takeaway was that I was able to take back a lot of knowledge that I could propagate to the scientists back home. And at the same time, I can say that now I have friends and colleagues in Canada, and this helped me develop my network. And having to experience a whole new culture in a whole new country sort of just broadens your view of the world and gets you out of your niche. And lastly, because this whole travel and experience was obviously by myself, my self-confidence and independence increased. And lastly, I just want to conclude by saying that it was a wonderful experience, and I strongly support and endorse research mobility, because it's not only beneficial for the research and the science as well as the educational aspect of it, but also you get to experience so many new things and meet new people. And so the social and cultural aspect of it is also extremely beneficial. And I want to thank people at Carleton, Yuan Clevers, and my professor, Dr. Alfonso Abizet, for giving me this opportunity and not only teaching me so many new techniques, but also giving me a good Canadian experience. Thank you.
Thank you, Gayathri. I like your point about introducing your kind of lab mates to your own culture by taking them to an Indian restaurant. I really like that point. And also you mentioned the weather being kind of new for you, which is actually a point that has come up a few times with people visiting, like during the symposium. So that's kind of funny. Our third presenter is Rodrigo Massera. Rodrigo has a bachelor degree in astronomy from 2014 and physics 2015 from the University of Sao Paulo in Brazil. He completed his master's in applied physics in 2018 at the University of Campinas in Brazil. In this project, he studied mammography image quality and dose optimization with Monte Carlo simulations and experimentally. Now he's in the last year of his PhD in applied physics at the same university. His project in advanced X-ray breast imaging with Monte Carlo simulations. Moreover, the project also encompasses machine learning for breast dosimetry. Last year, he came to Carleton University through the Emerging Leaders in the Americas program. Go ahead, Rodrigo.
Thank you very much for the introduction. So today I'm going to talk about the research project that was developed during my stay in Canada at Carleton University, which is entitled Investigating the Energy Deposition in X-ray Breast Imaging Using Computer Simulations from Cells to the Whole Breast. So on the left, there's a picture of me. Oh, sorry. First, I would like to thank my supervisors, Professor Alexandra, which is my PhD supervisor from University of Campinas, and also the Professor Rowan Thompson, which was my supervisor during my stay at Canada. So on the right, there is a little illustration showing the journey from the South Hemisphere to the Northern Hemisphere. Next slide, please. So before I talk about the project, I would like to talk about a little introduction and the motivations. First, ionizing radiations, in this case X-rays, are used in breast imaging. As an example, mammography, which is a widely used technique for breast cancer screening. Thus, a significant portion of the female population is exposed to X-rays. And there's an interest to actually study how the energy is deposited within the breast. However, it's quite difficult to perform this experimentally, and often computer simulations are used for this purpose. And what we are interested in those type of studies is the dose, which is the ratio between the deposited energy and the mass of the tissues. And we are mostly concerned about glandular tissue because it's the most radiosensitive when exposed to ionized radiation. And what is interesting is that the behavior of the energy deposition changes with the scale length. For example, we can study how the behavior is for cells within the breast, and we get a certain behavior. Then we change to a millimetric scale, and we expect another behavior. And finally, for the entire breast, we see another behavior, and they're all correlated. So it's interesting to actually study how they are related to each other and see how they influence each other too. On the right, there's a picture of the computer-generated breast models and also compressed breast used in those studies. And also included some example of the cell geometry that were simulated. This geometry encompasses around a few thousand cells with nuclei and cytoplasm. Next slide, please. And the actual development of this project was because it's unfeasible to simulate every cell in the breast. We actually select some regions of interest inside the breast and perform the cell simulations in those specific regions. So part of the project was to actually develop code to interact between this multiple scale of the simulations. And here are some results that I'm presenting. On the left, we are comparing the glandular dose distribution for different breast imaging modalities. On the top, we have the mammography imaging modalities, and on the bottom, the breast CT. The scale is using the quantity milligray, which is the quantity for dose. As we can observe for mammography, most of the energy is deposited on the top of the breast, which is compressed. And that's because of the beam energy and the geometry of the system. In the bottom, for the breast CT, the dose is much more homogeneous across the breast. That's because the beam is more energetic, and also the exposition geometry. The breast CT consists of several expositions to the breast, around the 360-degree images. On the right, what we are actually seeing is 3D specific energy distribution in cells. And for this, we selected small regions in this mammography radiation and actually simulated those nominal doses. Starting from 1 milligray, we observed that a lot of cells are not irradiated and the specific energy is quite low. As we increase for 5 milligray, we observed that more cells are being irradiated. And for 7 milligray, this actually increases. And then we get for 9. And finally, 11. So we observe that more cells are being irradiated. And what is interesting is that the specific energy is very generous. We observe that some cells receive energy deposition and some cells a higher energy deposition. And in this scale, as we see, up to 32 milligray of specific energy deposited in the breast. Next slide, please. Now I'm going to talk briefly about my intercultural experience during this project. First, I would like to say that it was my second time in Canada. First, I went to a conference in Montreal, a really beautiful city. But it was my first time abroad that I spent several months. and I must say that it was a wonderful experience for me. Ottawa is also an amazing city. I had this opportunity to actually meet wonderful people that helped me with my research, and also knowing the university and the city. This opportunity to share knowledge, I quite learned a lot from this experience. For example, I here in Brazil use a specific computer simulation program, and I had this opportunity to know another computer simulation program, to start learning about it. Also, it was my first time in another research group. It was a really good experience to know how the research is done in another groups, especially in this university, Carleton University. And on the right, there's a picture, a photo that I have taken that I went through during the winter. So for me, it was also this different experience because here in Brazil, it's actually 30 degrees right now. And actually being in Canada during the winter, -10, -20, it was an unique experience for me. But it was really, really nice. And I got this opportunity to go ice skating and those stuff. It was really good. So thank you very much for your attention, and please, any questions, I'll be glad to answer them.
Thank you, Rodrigo. That was wonderful. Yes, we can now open it out to questions from our attendees. If I can ask all of our speakers to maybe turn on their cameras. I will start, actually, I will go backwards. So I will start with Rodrigo. Rodrigo, I have a question for you. First, what interested you in this area of research, breast imaging? And relatedly, what made you choose Carleton for your ELAP internship in this area?
So, okay, really good questions. So I was mostly interested in breast imaging because first I was interested in going in the medical physics area at University of Campinas, where I started my master's degree. And I had this opportunity to meet my current supervisor, Alexandra, and it was her field of experience in breast imaging. So I started researching, and I really like it. This field is, there are really recently developments being made involving the images, the techniques, also with machine learning and AI. So it's a really hot area right now. So I think that's weighted a lot for this decision to start in this area. And for Carleton University, actually my supervisor met Professor Rowan in a conference in Italy. And she said about this opportunity with this program, ELIP. And I went and researched it, and Carleton is an excellent university. So for me, it was, I was really happy to have this opportunity. So yeah, everything come together, and so it was really nice.
Nice, that's great. I actually have a slightly follow-up question. So how has your research at Carleton advanced or informed your current research in Brazil and your like ongoing PhD work?
Oh, that's another good question. So originally, my PhD plan, the project did not had this subject about this multiple scale dose studies, and it was something that happened because Professor Rowan is actually studying those relations between different scales and the energy deposition, and we adapted for my PhD project. So it was something that came along naturally as we were discussing the project, and it really helped me a lot. It contributed for me, my PhD a lot.
Wow, that's great to hear.
That's really good.
Yeah, that's wonderful. Yeah, wow, that's great. I'm going to move on to questions for Gayathri. And so we had one in the chat that asks, what are the main causes of obesity that your research tries to address, and whether your research can be replicated not only in India, but also other regions?
Yes, good question. So basically what we're trying to do is try to see if a hormonal intervention can be developed for obesity. So since ghrelin is one of the hormones that's primarily responsible for appetite and fat accumulation, we thought that if we can clearly map out a way in which ghrelin affects female metabolism, then we'll be able to see whether we can interfere in any of these pathways to sort of provide a therapeutic intervention for obesity. And yeah, with respect to how this can be replicated, is it's not focused only on India. One of the reasons why I talked about how much percentage of women are affected is just to bring focus to the seriousness of the issue. But this is a general intervention. Obviously, the differences between different cultures and the environment in which people are living in. But ghrelin is something that is, ghrelin interventions are common across different cultures as well. Maybe the dosage and the kind of interventions can be tweaked for specific people in different lifestyles. But overall, our intervention was as generalized as possible for people from different countries as well.
And I'm curious, similar to my question for Rodrigo, because you did this work in your undergrad, the research internship, right, at Carleton was during your undergrad studies. So has this also informed your PhD work? Are you still working in the same area?
So it's not exactly the same area, but it has definitely helped me because at Carleton, I worked at a more neurobiology level, and right now I'm working at more psychophysiology level. And for me, I always believe that it's good to work from ground up. So it helped me realize, it laid a good foundation for me to branch out from there and continue my work in the same field, except just at a more psychophysiological level. So I'm not in the exact same area of research, but research at Carleton definitely helped me learn about the foundations of neuroscience, which is now helping me in my PhD.
Okay, nice. Thank you. So I'll move on to some questions for Alexandra as well. There were a couple in the chat. We'll start with, how has your research impacted your view on women's rights and reproduction?
That's a great question. Definitely going into research like this, you have to be very aware of your own biases, your own personal biases, your own personal perspectives and views, and even your own lived experience. It was hard at first. I definitely came in with some very strong views. I think it's helped me be more open to different perspectives. So during this research, we didn't only interview the pro-choice movement activists, but we also interviewed pro-life activists. So it was really interesting to get that other aspect, that other side of the debate. They're very reasonable, logical, human people, and they have very normal concerns. And there was a particular point in the research that resonated with me, and it was sort of this, when working with Together for Yes, that very strategic organization that I had spoken about, they did this thing where they created a middle space for people from both sides to come in. It was called this like gray area, this middle point where it wasn't as aggressively yes and aggressively no. So people who were maybe on the fence about it, but had some questions and had some concerns about the topic, they weren't really sure what the procedure looked like, they weren't sure who was getting the procedure. And so that created this sort of community in the middle where people could have differing views and perspectives, but they could also get along and they could also talk openly about how they felt and what they've gone through. And so it really showed me that you can have your own opinion and you can have perspectives, and they're most likely fueled by lived experience, but it's also okay to be questioned about them. It's okay for you to question them. why maybe this isn't the right way of doing things. Maybe your perspectives will shift over time. So it was really about self-reflection again, and I keep talking about it, and I think maybe because I have a sociology background, but I think it's really important to self-reflect a lot and to ask yourself those hard questions and to look to other people to help you answer those really hard questions.
Follow up to that, actually. So would you say then that your work in Ireland, like going there, being physically present there, was important for that kind of consciousness shift?
I don't think this research would have been possible without physically have been being there. Like I mean, maybe theoretically it could have been possible to do online interviews, you know, nowadays. Like I think I wrapped it up right before the pandemic happened. So I'm sure if I was doing it during the pandemic, we would have done Zoom all the time. But it was really, really important, especially the interview process, because when you're talking about a topic that had dire consequences for a lot of people and has really, really negatively impact a lot of people. But then there's also, you know, some people view this as a positive thing. They had a positive experience with abortion. And so it was really, really important to sit there with them and build that rapport and continually work with them. It wasn't a, let me interview you and then I'll never talk to you again. It was very much this relationship building throughout the 4 months. And I had the absolute pleasure of doing my very first interview on my own with the founder of the In Her Shoes Facebook group. And it was something I will never forget. So because I forgot my purse on the bus and we had to chase it down like we were in some action movie. But it was a beautiful, hard, and surprising experience to be there in person, to talk to so many different people and to see how this particular topic affects everybody. It's not that stereotypical image that most people carry with them, but it really impacts everybody. So I absolutely think it's essential for me to be there in person.
We had a speaker this morning who talked about the importance of the physical presence as being an emotional presence as well. And that is extremely enriching, right? That you don't get through this kind of more virtual experience. A question also I had for you, Alexandra, is because this is such a sensitive topic and coming in from outside of the country, was there, did that impact your research at all? Like, did you get any pushback? Did it matter for people that you weren't from Ireland, right, but conducting this research?
Yeah. So that's really great question. A lot of people were actually curious about that. I had given a presentation elsewhere. I think at some points it did, although the activist portion of interviews, they were just happy to talk about their experience. They were I've never experienced such passion before in the rallies, in the marches, even in the book reveals. There's a book out. It was. They were just really ready to talk about the movement. They were really ready for everyone to hear about what they were doing and the progress they were making. Also because the time that the referendum was repealed in the Republic of Ireland, it wasn't in the northern part of Ireland. So there was still that fight, even though it was over for the Republic of Ireland, it wasn't for the northern part. Mm-hmm. So it still felt very much like everybody was ready to fight for them now. And so having someone come and do research on this, because it was, again, a taboo topic for a lot of people in Ireland, they were happy about it. However, that being said, there was that, it was more so that I was an academic and a researcher. The pro-life side was very hesitant. They weren't sure how I was going to use the data, the information I was gathering. They weren't sure how I was going to portray them or their side of the movement. And so I did get a little bit of hesitation there, but I continued with the back and forth relationship building with them. And that was really important so that they could see every step of the way that I wasn't going to villainize them or their movement or their thoughts or beliefs. So I think that was the only part I got some from.
Mm-hmm. There's another question. Were there any instances where you encountered individuals who had moral conflict with your research?
That's a good question. So I should have given it. I mean, it was written in small at the beginning of my presentation, but we didn't. So we got ethical clearance from Carleton because, you know, it's a very sensitive topic. There are some people who have been victimized, and however, we did not. So the stories of loss were gathered indirectly. So we didn't actually interact with any vulnerable people who had gone through something traumatic. So we didn't really get any resistance that way because we mostly talked to activists, scholars, university professors. We talked to legal representatives. We talked to some political leaders. Mostly we got, which I was also surprised, I expected some pushback, but we got a lot of positive reinforcement and being told that this is a really important issue, especially for Ireland, and that it needs to be pulled objectively as objectively as it can be. I will recognize we're 3 women who were researching this topic, so there are some inherent biases going on. But no, it wasn't, there was no real pushback. We did witness some protests happening outside of hospitals. There were a lot of online angry comments, you would maybe even say hate speech. So yes, there was some of that, but never targeted towards us. And we didn't have anyone upset during our interviews or anything.
Okay, that's good. Sorry, I know it's 3 o'clock, so it's kind of the end of the session, but maybe if we can hold on for a couple more minutes. I have one question left maybe for Gayathri, and then I will wrap things up. So Gayathri, is your research now still being conducted with Carleton?
The continuation of the project?
Yes, I guess, do you still have any connections with Carleton in relation to your research? And do you know if Abizaid's lab is continuing that kind of research? I guess I'm going to break this up into 2 different questions.
Yes, so I'm still in touch with my colleagues at the Abizaid lab. And as I mentioned before, I was able to finish only half of the project. So the other half is being continued by people at the Abizaid lab. So I am in touch with them to sort of help them analyze the data and help them bring the project to a complete loop. So in that sense, I'm still connected to Carleton.
Okay, great. Well, thank you so much. I'd like to thank all of our speakers. They were very, very interesting presentations. And also thank you everyone for attending the symposium today and attending this session. We heard from so many different speakers coming from such a diverse range of experiences and disciplines, but all benefiting so greatly from an international and intercultural exchange. Obviously, there is a strong interest in making international connections in research and collaborating across borders, as we see with the attendance at the symposium. We've had, I think, 200 attendees register, and this is very exciting as we look to the future towards hopefully further mobility when we can, and more collaboration across borders, and actually being able to move and connect after the pandemic. So thank you all for coming, for joining us, and thank you so much for the presenters. They're really interesting research, and it was really great to hear about your intercultural experience and your experience at Carleton, or in Alexandra's case, your experience abroad, and how much it informed your research and your thinking, right? So thank you so much.